In this lecture, I'm going to talk about the circuitry of the retina. I'm going to make a few assumptions as I go through this video. The first is that you already understand phototransduction. I'm not going to talk about that. I also assume that you understand the difference between scotopic and photopic vision, so I'm not going to talk about that. I'm also going to assume that you understand that photoreceptors emit a dark current, that is, that they hyperpolarize in response to light, and that light does not depolarize them. So the first thing we're going to look at then is the overall design of the retina. It's quite striking that if you look at the retina, you can actually see these different layers as you take a section of the retina. It doesn't matter about the details of these for the moment, because what I'm going to do now is to simplify that representation for you so that you'll understand the circuitry rather than the anatomy. Here's the overall design, and just so you can see who this attribution is. We have at the top, if you like, which is the bit nearest the light, the light comes down this way. We have a ganglion cell layer, an inner plexiform layer, an inner nuclear layer, an outer plexiform layer, and an outer nuclear layer. So let's just make a quick copy of that so that we can comment on it. So the important points are this inner layer here and the outer. So if you focus on these just for a minute. So the key players here are the photoreceptors, which we have down here. So the rods and cones. If you just ignore some of these cells, but just start with the photoreceptors, the next step is the the bipolar cells. And there's two types, rod and cone bipolar cells. And then they go up into the ganglion cells. So that's the main design here. So I'm going to simplify that with a little diagram. So we start with our photoreceptors. Then we have the bipolar cells. And they feed into the retinal ganglion cells. So remember, light is going in this direction and information is going up in this direction. So the photoreceptors pass the message on to the bipolar cells pass it on to the retinal ganglion cells, and then the retinal ganglion cells, their axons go up through the optic nerve. So this is your basic design. That's your basic through design, but we've also got to add a couple of other things. So let me just get rid of this light up the stuff on the left. Let's draw certain. We've also got on top of that horizontal cells and amacrine cells. And they pass information from side to side. So you've got this basic arrangement. And of course this maps out pixel by pixel the receptive the image on the retina. And so these, you've got a flow going up and out, but also a sideways flow of information this way around as well. The other thing to be aware of is that the fovea is adapted for high acuity vision. So if you imagine you've got the whole of the retina, you've got an area in the middle called the fovea, and that's close to where all the axons merge and then go out. And this area, it's adapted because it has, first of all, a large number of cones per unit area. So that's a bit like having a camera with very small pixels. If you've got lots of pixels packed in a small area, you've got a good camera image. And that correspondingly means they've got small receptive fields, and they're also adapted for fast responses to light. So that's how the fovea is adapted to high acuity. Next thing we want to look at is the synapse from the photoreceptors to the bipolar cells. So we're now going to focus on this synapse here. The first thing to say about this synapse is that it's mediated by glutamate. So you've got the photoreceptor, whether it's a rod or a cone, they all release glutamate. The bipolar cell can respond to this with excitation or inhibition. You might know that glutamate receptors have two types. There are ionotropic, where glutamate always results in excitation, but you also have metabotropic, and depending on which type, you can have excitation or inhibition. So, if we have ionotropic receptors, remember glutamate, if I add glutamate, this will result in an excitation. But remember that glutamate is actually released not in the light, but in the dark. So if I have ionotropic receptors, the bipolar cells will respond to light being off. Conversely, if I have a metabotropic glutamate receptor, which is inhibitory, now the bipolar cell responds to light. So we've got two types that we could possibly have of bipolar cells. You'll see in a couple of minutes why that's important. So let's look at the answer to that question now then, shall we? So what this means is that there are actually two types of bipolar cells. We have on bipolar cells and off bipolar cells. What that means is if I'm recording from a bipolar cell, and this bipolar cell is getting input from a photoreceptor, and what I do is if this is the light level I'm plotting here, if I turn the light from off to on, in other words I just shine a light onto this on bipolar cell and I measure its voltage, what it does is it actually responds with a depolarization to the light. It doesn't have action potentials. This is the interesting thing about bipolar cells. They're all graded responses, just like with the photoreceptors. With an off bipolar cell, if I have the light on and switch it off, now the off bipolar cell responds like that. So we've got on bipolar cells which respond to light on with a depolarization. We have off bipolar cells that respond to light off with a depolarization. So they're actually doing the opposite thing. And that's because, I need to think about this, think this through now. So if you've got light, so let's imagine it's dark here. Remember, the photoreceptor is releasing glutamate in the dark. So when you switch the light on, we're getting this response. The only way you can get that if this is actually an inhibitory synapse. Let me just explain why. During the dark, actually it might be clearer if I just make this a bit, so light, dark. Okay, so during the dark, we've got glutamate being released and the membrane potential is negative. During the light, we've got less glutamate released, and because this is an inhibitory synapse, less glutamate means less inhibition, so it actually goes up. It's the opposite here. Here, I've got light and dark. This is an excitatory synapse because, remember, it's only when in the dark they get release of neurotransmitter. So the neurotransmitter is being released here and we get excitation. So it might help if I draw a line where a yellow line where the neurotransmitter is being released. So the neurotransmitter is being released here, and that's causing the membrane potential to go down. So that's inhibitory. Here, the neurotransmitter is being released here, and that's causing a dipole. So that's excitatory. So now let's make things a little bit more complicated. Let's have a look now at the receptive field of a bipolar cell. So imagine that I've got the whole retina like this. So this is your whole visual field, and I've picked out a bipolar cell right here. Okay. Now remember, we found an on bipolar cell. So now what's going to happen is I'm going to take my light and I'm going to shine it on the bipolar cell like this, and what I see is the cell depolarizes while the light's on. That's why we call it an on bipolar cell. So this is the light here, I switch the light on and I get a response. Now something funny happens